Hello guys. Hello guys. Today now I'm going to discuss one small lecture, one tutorial video. This is not pertaining to any particular course. This is about one basic concepts, that is micropropagation. As horticulturist or agriculture scientist, agriculture students, we should have a basic idea of those things. Okay. So what is this micropropagation? Before going to micropropagation, we should know about propagation. Okay, my handwriting is not that much good in system, but you people can adjust. Okay, so come to topic. Propagation means multiplication of plants. Okay, we know we have to multiply for growing any crop commercially. We should go its multiplication. So how this multiplication happens? Okay, naturally plants used to multiply, that is by sexually or asexually. You all know about this sexual propagation, how it happens by pollination, fertilization events happens. As we know little bit in our basics plant biology, we have studied these things. But what happens actually in sexual reproduction? As we know, when male gamete and female gametes, they both fuse together, the zygote used to form, and it will go to embryogenesis. Embryo will be formed, and that embryo will develop into a full plants. Okay, so this happens in case of plant as well as animals. In plants, another one important aspect is there in propagation, that is asexual propagation or clonal propagation or vegetative propagation. Means what? Any plant cell, we are taking it and we are propagating it, means it will grow to a new plants. That plant cells has a specific features that is known as totipotency. Means the you people can study more about stem cell. Stem cell, in case of animals, the stem cells are found, and stem cells have the potential to develop a new cell. But in case of plants, only all the cells have. In case of animal, only specific stem cells are there, like bone marrow cell and other cells are there. But in case of plants, any cells can have the potential of stem cell, but we have to explore that potential. Okay, so that happens. Or some specific vegetative propagules are there's like bulbs, tube, no, in case of tuberose, we used to get bulbs, bulbils. These things are vegetative propagules. But in commercial propagation, we have developed, we have explored this thing through different technology, vegetative propagation technology. For example, grafting, budding, layering, and several micropropagation tools. Okay. So we'll discuss about this micropropagation, what happens, or tissue culture. It is otherwise known as micropropagation or tissue culture. In many of my lectures, I have discussed these things in classes, but still I want to um, refresh your minds. Okay, so be attentive. This is a casual lecture, and it is, and you people can refresh your minds through these things. Okay, we'll see. So the word micropropagation, again we'll see, sorry, micropropagation or tissue culture. As this word implies, micropropagation, we are propagating in from a micro object, or you can tell it like from microscopic, not exactly microscopic, small plant tissue we are developing the plants. Or this tissue culture, what do you mean by tissue cultures? Any tissue, when it is being cultured and a new plant is being developed, any tissue, remember this is nothing specific, any plant, any tissue, when it is developing, that is tissue culture. Okay, so how it happens in case of plants, this is quite interesting. In animals also tissue culture are being there, like in vitro fertilization and other things are being explored day by day, and it is being used. In case of plant, it is quite useful. First, you remember, I forgot to mention one important point during that sexual and sexual propagation, that in case of sexual propagation, when male and female gametes fuse, the progeny will be formed. And since the chromosomes used to the crossing over happens in case of meiosis, so what happens, that characters changed. Okay, remember this thing, characters change, it segregate, the new characters are developed, okay, by introduction of this male and female group. But in case of vegetative propagation, nothing happening, mitosis is happening, somatic cell division is happening, just we are taking the tissue, it is growing. Okay, so that is the basic difference. So in case of micropropagation, the micropropagation or tissue culture, this can be, this is also one kind of vegetative propagation. Remember this is one kind of vegetative propagation. Okay, so because we are taking from vegetative cell, even though we are taking from reproductive tissue, still vegetative propagation happen, but few cases are there, I will explain when I will come, that time we will know here also how crossing overs and those things happens. Okay, so first we'll discuss the types, how it happens. You just remember, micropropagation or tissue culture means from a small plant tissue, any tissue, we are developing the whole plants. Remember this, from small plant tissue we are developing the whole plants, just by in vitro condition we used to do this, and by modulating the hormonal balance. Okay, auxin and cytokinins, remember these two are the important hormones in micropropagation, auxin and cytokinin, these two. And the ratio between auxin and cytokinin. Okay, remember this ratio between auxin and cytokinin. When this cytokinin, auxin to cytokinin ratio is high, it will form root. Remember this. Okay, when auxin to cytokinin ratio is low, it will form shoot. This is the basics, this is the basic concept of micropropagation. Remember this, when auxin to cytokinin ratio means auxin will be high, cytokinin will be low. This ratio is high means what, auxin will be high, cytokinin will be low, then root will form. When auxin will be low and cytokinin will be high, shoot will form. Example I can give you, in tissue culture, when we'll put more cytokinins, like the extensively used cytokinin is kinetins or BAP, benzyl adenine phosphate. It will help shooting in media we used to put. Okay, then auxin, one example, these are examples I'm giving. Example is IBA, when we'll put, it will put rooting. So we'll see what is happening in micro propagation. There are two kinds of micropropagation. Okay, so one is direct organogenesis or indirect organogenesis. I told in micropropagation means from a small plant tissue, in tissue culture, from a small plant tissue the whole plant will regenerate. Okay, the tissue will regenerate. This feature is known as, this feature of plant cell is known as, sorry, totipotency. Totipotency. Okay, totipotency. This totipotency is a characteristic feature of plant cells. Okay, because of this, the plant's single cell can form a complete plants with shoot and root. Okay, so this is this what happens. Then next one is, I was telling there are two kinds. One is direct organogenesis, indirect organogenesis. Okay, so when we are taking a differentiated plant cell, remember this, when you are taking a differentiated plant cell, the two cases are there. Okay, direct organogenesis, indirect. In one case we are taking differentiated plant cell, and in another cases we are taking de-differentiated plant cell. So you remember this. um, Before coming to these things, so I will tell about, just I will brief about the cell differentiation. Do you know what is cell differentiation? And I think you people know these things. In this is kind of one-way communication is going on. But if you people want to give your comments, you are always welcome to put in comment section. But okay, what is the cell differentiation? Cell division, you all know. Cell division means cell will be divided. It will be multiplied. Okay. There are two kinds of cell division: mitotic cell division, meiotic cell division. You know, mitotic happens in somatic cell, meiotic happens in zygotic cells. Leave it. Differentiation: when the divided cells will be assigned to a specific tissue. Okay, will be defined to a, well, it will be given in a specific tissue. When it means tissue, the cell is becoming tissue means one specific assignment is given to that tissue. Okay, when you take the, you can take the example of meristem. Okay, in class also I used to give this example like murgi kya jane ande ka kya hoga. Okay, you would have heard this is a famous song in movie Three Idiots. मुर्गी क्या जाने अंडे का क्या होगा, लाइफ बनेगा या तवे पे फ्राई होगा. We don't know what is happening. That is the fact happen in case of somatic tissue or meristematic tissue. When meristem is dividing and it is multiplying, the simple cell mass meristem, you know, this used to be represented as like this, then like this. So this used to divide, grow like this. Okay, it doesn't know what this cell, they're just multiplying, they don't know what work they are going to do. Okay, so when it will be given, the complete cell will be given to a specific assignment, you cell have to do this work in a, and you have to form this tissue, you have to do this work. Suppose once it germinates, the epicotyle part will be given work that it will grow as a shoot, it will, leaves will come, photosynthesis will happen, then the assimilates will be distributed throughout the plants, it will grow and it will favors the growth and development, fruits will come, this happens. And the hypocotyle part, this, remember this seed germinate, this is epicotyle, this is hypocotyle, this will become shoot, this will become root. Here there's differentiation started. Okay, root means they have to absorb nutrients, they have to supply the inputs, that is the thing. Here oxygen, here cytokinin will be formed, different things are there. Okay, so this differentiation means different assignments, the cells will be assigned with different assignments. Okay, that is differentiation. So now I was telling about direct organogenesis and indirect organogenesis. First come to direct organogenesis. So when we'll get the de-differentiated mass of cell. Okay, this. So this is direct. Okay, when we'll get shoot tip. Okay, remember the shoot tip. So there will get a de-differentiated mass of cell. The cells, they have to grow, they have to become the shoots. Okay, shoot tip means the shoot will be formed. Shoot tips, root can form in plant by nature. Okay, so we'll take the shoot tip, we'll put inside media, control condition, so obviously it will form as a shoot. Okay, we'll put the BAP concentration because BAP favors the shoot formation and it is the aerial part formation. So we'll put a cytokinin, so it will favors the shoot elongation and shoot will be formed. That is happening. Then when a specific height will be achieved, or we want to multiply, we'll go for subculture, then we'll develop into small plantlets, we'll multiply, more number of plantlets we'll get, then we'll put it in rooting media, then root will be formed, then we'll can get the complete plants. That is happening direct organogenesis. In case of, in other case, okay, indirect organogenesis. Indirect, in case of indirect, any differentiated cell. Okay, uh, I will write this again. uh, Direct, here d differentiated tissue. Indirect differentiated, you just remember this 2 point, it will be easy for you people. Indirect, what happenings, we are taking any tissue like leaf tissue, root tissue, flower, petal, anything. When it is already one specific assignment was given, but we are taking it, for example, like in case of emergency. Okay, one officer, he's posted in some block or something, government will take in that no, your assignment is now, you no need to do that work, you have to come like one agriculture officer. Okay, so but that emergency, some you can take this example like election time. Okay, that time all the officers will be pulled together from different allied departments, and they will be all will be diverted to election to conduct election duty. Okay, so like that, when one what happens that officers will be taken by the government and they will be assigned new functions. So for that, first they will be instruction should be given. So what happen, suppose we are taking one flower petals, okay, and we have to develop plants. You know, totipotency is there in plant cell, I was telling this now. So what happened, when we'll take the plants, we have to make it to original form. Okay, we'll take, and then that time after taking them from the from that already assigned work, they should be given to neutralize their all work. They should forget that what I was doing this that, but they have to, because they should do new, they are going to do new assignments. So that thing is known as this de-differentiation. Will do the differentiated cell will be differentiated tissue will be done. This de-differentiation will happen. De-differentiation will happen. Then again it will go to re-differentiation. Okay, re-differentiation. First, that already differentiated thing will be de-differentiated, neutralized. Then new assignment will be given. Re-differentiation, that is indirect organogenesis. How this new assignments given, how this de-differentiation happen practically. This is theoretically I told. What happen? Suppose we are taking, so that will be callus will be formed. One terms new here then callus. What is this callus? Callus is, so I explained what about this differentiation. So callus is undifferentiated. This is undifferentiated mass. Okay, undifferentiated cells. So this is undifferentiated because already it was differentiated, but we have de-differentiated. Means undifferentiated mass is formed. That is callus. We want to form, we want to make root from callus. Will balance the oxygen cytokinin ratio. I was telling for rooting oxygen should be higher than cytokinin. Then we'll put more oxygen, obviously roots will come. But first, generally what happens in case of tissue culture, first we'll generate the shoot part, aerial part, then we'll go for rooting. But root will come, they will absorb nutrient. Then what will use, who is going to use the nutrients? So if shoot is there, then root is forming, it will be used. Okay, so the first we'll put, after this dedifferentiated cell will get callus, we can put them in, high oxygen media, sorry, high cytokinin media. Then the aerial part will be formed. Then after having a certain height or certain number of suits, we can multiply the suit. Suppose from this is one callus, we are making one suit, we want multiple suit, we'll put in multiplication media. There are different hormonal concentration for different plant, we'll get different plants, will subculture will separate each plants, and in each test tubes we'll put them, then each will be rotated, so more number of plants will be generated. So this happens in indirect organogenesis. So in today's class, I just brief about propagation. What about propagation? Then asexual propagation, sexual propagation. Then in asexual propagation, different things are there, ducting, budding. We discussed about micropropagation. This is one kind of asexual propagation. In my micropropagation, I discussed what happens exactly, the basics of micropropagation. Then what happens in direct organogenesis, indirect organogenesis, how these things happens. Then in indirect organogenesis, we'll see the different. Um, I told about callusing, how from callus suits developed. Then different types of cell cultures are there. Actually, we'll discuss in next class what are different types of direct organogenesis and what are different type of indirect organogenesis. So hope you people have understood today class. You have enjoyed this lecture. You can put your doubt in comment section. Thank you for watching. Thanks.